Hello. Oh, connected.
Hi, how are you?
Hey, how are you? Pretty good.
So I cannot hear you. Hold on a
You cannot
second.
hear me?
I can hear you now.
Okay. Let me just. Oh, okay. So, how's life in Toronto? I saw that you moved to a company now.
Yeah, I moved to a company, but this is still in Toronto.
Mm-hmm.
It's called Phenomic AI. Still single cell analysis for cancer. But yeah, it's a startup.
Good.
Hmm.
Exciting. Always exciting.
Yeah, changes are always exciting and going well so far.
And the AI part is deep learning on the image data, or?
Uh, we have a component based on imaging and another one based on single cell RNA-seq.
Oh wow.
But yeah, it's because of machine learning.
Mm-hmm. Cool.
Where are you located again?
In
France?
Paris, yeah, or close to
In
Paris.
Paris.
Mm-hmm.
Okay. How are
France.
things going with the restrictions?
So we have a lockdown at 7 p.m.
Mm.
Kids are going to school, half day, or yeah, it depends on the age, but between half day and full day. And otherwise, no sports, team sports so far.
Hmm.
Golf courses are open. Mm,
Okay.
yeah, it's. I have the impression it's getting better actually. I hear
That's
it's not
good.
as bad as in Canada. I think we've been through our third peak here.
I see. Yeah, here in Toronto, we're in the third wave as well. Still some restrictions, but the rollout of the vaccines is getting a little bit faster now. So there is some hope at the end of the tunnel.
Yeah, we'll see. If there's no mutation coming from anywhere.
Oh yeah, man! Don't tell me about that. Might take forever to get, but at least people are not dying that much now, so that's good.
Yeah. But it's good, right? Without the pandemic, we wouldn't know each other. So
That's right. That's right.
there are a few light spots around.
Yeah. Do you want to start trying to share your screen just to make sure that it works?
Sure. Let me see. Um,
Excellent. I can see your
okay.
slide.
I guess I move this over. And I know, probably not closing.
All right. I think we can take it over.
Okay.
Thank you everyone for joining today. Today we have Bernd Hagla,
Jagla. Ouais.
Jagla, who has been working on Tempora integration for single cell time series data analysis in a Shiny application, working with different aspects of the analysis. Thank you, Bernd, and take it away.
All right, thank you. And thank you for the opportunity to present what I'm doing. Let me see if I can start this presentation. I hope you can see this. I'm just going to quickly introduce the group I'm working with, and then I'm going into medias res and describe basically what this Shiny application is doing and how the integration with Tempora is working. I guess you all know Tempora, so I'm not really providing too much information about this, but anyways. So I'm working at the Pasteur Institute in France, and for the last, I guess, 11 or 12 years now. And it's I'm working in a core facility there, which is called UTEX, yeah, cytometry and biomarkers UTEX. It's in the end, it's a core facility. And so we are working basically with all of the departments here that are listed as rows. And then these departments are connected via a Center for Translational Research, which is also connecting us with the hospitals in the area. And then the Center for Technological Research and Resources, which is basically a collection of all the core facilities in Pasteur. And so everybody has to be associated to a department, and we are historically based connected to the immunology department. And I don't know if you know this, but the Milieu Intérieur is a large case study on healthy human donors, and so this is basically where this all evolved from. And oops, so in the end, what we are doing is we are taking in human or mouse samples. I don't know how we can stop this. Okay. That is not nice. We get to this afterwards. So we have many users, almost 270 users, many projects. Most of the stuff we are doing is basically with the immunology department. That's another reason for our affiliation. And since we are also working with human patients and in the P2 Plus lab, we have to have different sources or different approvals for the experiments that we are providing to our users. And so that's why we had to invent our own project submission tool, which is called Maven version 3 now. And so this is taking care of all the administrative part and coordinating the approval of all the necessary entities for each experiment that is performed with us. And so this is a group. We have by now 2 bioinformaticians. Milena Hazan is the head. And so logistically, we are divided into the biomarkers unit, which is mostly RNA-seq and single cell nowadays, RNA-seq based approaches. And then we have the cytometry platform. And they host a bunch of high-tech or high-end cytometers, and then we are now starting also immunomonitoring platform. Yeah, but this is really in the very beginning. And so we have some 32 technologies in 6 laboratories, 2 BSL 2 and 3 levels, and yeah, quite a few material. And so I'm responsible. I was the first bioinformatician there, and I And I mostly working with single-cell RNA-seq data and cytometry data. And since our new bioinformatician has joined, I am now trying to focus more on the cytometry part. And let me see. Right. So so this is basically getting me into the SCHNAPPs business, which is this single cell shiny up for single cell RNA data analysis application. And the reason for developing this, this was I think 3 or 4 years ago, there was no tool available for this. Now there are about 80 or more tools that do similar things. And back then we decided to develop our own tool because the researcher really has to do most of the cell selection and data analysis in terms of understanding the individual cells. And so I don't, or I cannot do this because my biological background is really not sufficient for so many, coming from so many different areas of research. And so that's why basically we developed this Shiny app. So this is the great advantage, or the great difference is here that you can actually select the genes and the cells that you want to work with. And the reason for this is that I believe that removing noise from a data set is very important to get much better data. And noise can be in the single cell area can be actually cells that you're not interested in or genes that you're not interested in. And the more you remove of these genes and cells that you're not interested in, the more concise and better will be your results. So that's my philosophy, and that's basically where this comes from. And so it's a Shiny app, dashboard design with the the functionality on the left-hand side and the visual part on the right-hand side. And we have this nice little logo. There is it's on GitHub. It's freely available. We have, because of the resources, compute resources that this can require. We have for our customers at Pasteur. We have a dedicated computer with 64 gigs of memory, and since memory is the most limiting factor, at least for us, and we can then deal with up to, let's say, 10,000 cells and in a human genome. So there, as I said, there's a lot of documentation out there already, including some short videos. Then there is something called contributions, so you can add actually functionality to this, and that is relatively easy and straightforward. So if the functionality is quite easy to adopt, then it takes only a few hours basically to really integrate this into the application. And so before going into the integration with Tempora, because this is really what I wanted to use this group for in the end, since you're in Toronto and you, or at least here in Gary's group, developed Toronto, I wanted to show off a little bit of how this kind of collaboration or this integration is working, and want to show you basically what I can do now with your tools. But before I do this, I quickly emphasize a little bit the functionality that I deem interesting, or at least a little bit different to other tools that I've encountered so far. And obviously, I don't know all the 80 tools, and I sporadically look at a few of them occasionally, so I might be completely off here. Right, so in the end, Schnapps can do almost anything. It can do the Seurat pipelines, the SCADA workflows, and normalization, clustering, and so on. And so this is an example for the QC, for some of the QC displays where you have the UMI count and the feature count. You can then change the colors interactively, go into density plots. You can, for example, if you want to select cells based on expression of a given gene, you can sort them based on this expression and then select the cells within this plot. So that's, for me, it's the easiest way to actually select cells. And so, right, so this is for comparison with well, any of the other without GUI workflows, where you select features, cells based on minimum number of features or counts. You can do this basically manually now without these thresholds, but you can apply any feature that you're looking for. And so you can zoom into this and then sub-select within this zoomed area as well, because we are using a Plotly functionality, and so anything that Plotly is providing, we are doing here too. And so this is an example of how one would select cells with a high mitochondrial count. You have the percent of mitochondrial RNA, and sorry for this mess with the slides. And so you the red cells are the selected ones. You then can select these cells individually by copy and pasting the names, and then copy them over to another panel where you actually input the cell names individually to be removed. And so there are a lot of kind of this type of interactivity there. Then, so this is a list of all the normalizations we have. For the example today, I have preloaded the thyroid object that Gary's group has provided, and so I can also do this. Basically, I can load in data that has been previously offline or analyzed or calculated. And so then for the principal component analysis, you have these dim plots and all these access to the loadings, the variable features. Most of the parameters are available for any of these normalization types, clustering, PCA. And I'm just, because of the PCA is very sensitive to which type of algorithm you're using, and to be able to really reproduce the thyroid and the SCADA workflows, I actually implemented both of these PCAs algorithms in there, so to be able to reproduce the results. Right, so now this type of heat map allows you to do this cluster versus all cluster, or any cluster versus all the other clusters comparison, where you can then ask for the direction if you only were interested in upregulated or downregulated genes, minimum fold change, and the number of genes per cluster that you want to access. And so you have a lot of freedom for choosing and modifying your heat map. You have, for example, you can set the maximum minimum value to adjust for the coloring here. This is quite useful sometimes when you are interested in the low range or high range, and you want to basically focus in a certain area to really visualize the differences between genes or groups of cells. And yeah, so you can sort and display any of these, change the size of the image and so on. And then of course you can save this to an RMD file or download the plot data for other uses as well. And so these are the this is a list of all the clustering algorithms that are currently being implemented. Yeah, I guess the cell ID is the most obscure one. This is developed at the Raoult lab, at Imagine, which is also in Paris, and so I am supporting our local French business here as well. Yeah, so this is just another example of a density plot. Not much to say about this. There's also a way to visualize in 3D, and here you have to acknowledge that you can change and select the axis as you deem or find interesting. In this case, I have PC1 and PC2 now, and the sample names are now on the y-axis, which or z-axis in this case, which allows me to actually visualize by turning the image now really which cells and how many cells are overlapping each other, right? I mean, if you look on from the top of it, the blue cells will cover the green cells, and you don't really know if they are overlapping or not. And so here now with this view, you can actually visualize the who is overlapping and which group is overlapping with whom. This is just one example of where I find this interesting to not only look at PCs from a PCA, but integrate other projections. And yeah, so Plotly also allows you to remove certain factors from the plot, which then allows you really to see which cell type in this case is overlapping with which other cell types in this UMAP projection. Histograms are quite useful as well. So this is just one type. If yeah, you can say you can select histogram as a y coordinate, and then it will plot you the estimated distribution, and the coloring is then according to these factorials here, and again you can select or unselect these as well. Then there's this violent plot. So there are many violent plots which allow you to look at different aspects of a given data set. I'm going to show this, I hope, in the example of Tempora later on. And so here now we are looking at two different factorials, and this is a very nice view for me to be able to select a combination of these factors. So for example, I only want to look at the endothelial cells from these clusters. Now I can actually really select these cells directly here, and I'm not confounded with any overlapping distributions. Okay. And yeah, so for me everything is a projection, right? Anything that is where you can develop a value per cell for each and all of the cells. This is a projection for me. This is the can be the principal component 1, 2, and 3. It can be gene expression. It can be the sum of genes over all the individual cells. And now I can work with these projections in that I can rename them, combine them, rename the levels, and so on. And so this is quite useful when you really want to rename, for example, the clusters that you identified that are originally 1, 2, 3, and now you want to name them, I don't know, B cell, T cell, or whatever. Okay, and so here, right, so this is one of these examples where I have the combined expression of CD8 beta 1 and CD8 alpha versus the CD3 gamma, CD3 delta, and epsilon expression combined. And now I can actually look at the co-expression of these genes and yeah, do some kind of cytometry equivalent of gating these cells. And obviously you can import the other expression values from, I don't know, from site-sec data or whatever, and then do the same thing there. Then one of the key questions I'm usually interested in is I know that a certain number of genes is interesting with respect to my biological question. And now the question is, are there any other genes that behave similarly or not? And actually mostly similarly to a given gene. And a lot of these tools and functionality that I'm providing is actually around this approach. So for example, here I can select a bunch of cells arbitrarily, and then I can actually look at the detailed information for these genes. So I can now specify another list of genes and look only for those cells that I've selected at heat map. I can look at the coefficient of variance or correlation coefficients. So this is depicted here. So here we are looking at the coefficient of variance, and we can sort this. By this we can now restrict this list to have a minimum expression, and a certain number of cells within the selected group have to have the genes expressed. And now I can look at these the genes that are basically co-expressing. And similar for the correlation coefficient. Now we have it calculates all the combinations of projections and gives me the p-values and corresponding correlation coefficients. And then obviously, or not obviously, but naturally I can select the cells, name them, and kind of group them together, and yeah, get the number of cells for this. And so this is then a new projection in the end, which is consisting of yes and no, or true and false values for each of the cells, and I can afterwards combine these selections as well to new factorials. I can, well, there's a differential gene expression analysis module where I can sub-select the input, basically. So I only want to look at the cardiomyocytes in this case. And I now, in the t-SNE projection, I now can manually select the groups of cells that I want to compare. With a relatively large list of differential expression algorithms, and then I get my volcano plot and a table with the differentially expressed genes. And yeah, so the usual, I mean, the table is changing depending on the algorithm that is chosen, but you get all of the relevant information. And now to finalize this very short overview of what Schnaps can do. Schnaps can also be easily extended, as I mentioned earlier, and so these are the extensions that I have currently integrated. I have a self-organizing map to identify similarly expressing genes. I have cell ID, as I mentioned, which is extracting signature genes and also clustering the cells. Scorpius, LPgraph, and Tempora are trajectory inference based projects. Scorpius is, I think, one of the earliest or earlier trajectory inference algorithms that was implemented. It has some very nice visualization features, and the size lab is, I don't know, somehow a befriended lab. So similar with LPgraph is coming from Curie, also Paris. And now with Tempora, we are expanding our horizon to Canada, and specifically then Toronto to basically cope with the demand of one of our collaborators who wasn't really satisfied with the current trajectory inference algorithms, and so he also has some real time dependent data sets, and he is also wants to identify, or at least identify the order of the clusters, which really fits with what Tempora is doing, and we are currently testing this on his dataset. Okay, so Tempora, now I'm feeling pretty stupid in terms of trying to explain to you what Tempora is. But yeah, it uses the information from the clustering and a provided pathway database in the end, and uses Arachne to really come up with a network within between these clusters, and then it orders and builds these relationships between the individual clusters. And since, I'm really not feeling comfortable to explain the authors of the tool, what they have done, I am moving on and leave these questions. I leave this for questions. So we have, or I have implemented the main features of Tempora, meaning the clusters. Now I can, this is interesting. Actually, let me try to switch to the live version. Can you see this? Can you see the application now? Or are you still seeing?
Yes.
Yes. Okay. So here, this is really a live version of this, and right. So here you have the clusters. You can actually select any cluster that you deem interesting. You can also select any of the variables or projections in the end, or factorials that are factorials as a time variable. And then here you can order the samples in this case, or the levels of the factor as you wish, and you select the pathway database, and then you click on apply changes, and then it runs. It gives you the the variance plot of the PCA enrichment analysis. You should select a value that is below or that is within the range here. So if you select 8, then it's not going to run. And then you get the network, basically the connection between the individual clusters on the with this time scale. And in a different view, you can now hopefully. Okay, I didn't run this earlier, so this takes a minute. But we can go on here. You get the individual pathways with the corresponding p-values. You can select one of them and get the expression of the clusters versus the inferred time. So here you really can now see if it's if the pathway is enriching over time or how it's behaving basically. And then you can plot this in any two-dimensional space that you deem interesting, and you can then see the connection between the individual clusters, in this case on this projection. Yeah, and so that's basically the integration of Tempora. What you can do now is once you have identified the pathway, in this case, I don't know, here the synaptic vesicle budding, blah, 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 then you can identify the genes that are corresponding or connected with this pathway, which are these genes, and you can use these genes to for in various visualizations within SCHNAPPs. And so in this case, you are now plotting the expression in the violin plot, and you see that it's actually also in the violin plot increasing over time. And you can now, well, this is the same thing, only that this allows you to actually require a minimum or maximum number of log transformed values basically to be expressed. So here in this case, the values of a cell is counted as 3, for example, if it belongs to the E11 sample and it has 3 of these 4 genes expressed with a range of expression between these two values, right? And so now you can change this minimum value and see if there's if the expression is really higher, or if there's an expression level is changing over time in this, or over the clusters. And what is also quite interesting is to see which combination of these genes is expressed, right? And so this is done with this list of combinations here, which are calculated on the fly. And now you can actually see the progression over the time points and how this is in this case, which individual genes are contributing to this. I don't know, it looks like the same. Now yet another view on the same data is looking at the combined combination of 2 factorials. So here we have the clustering and the time points, and we can see now over the clusters how the time points evolve within each cluster in the end. Okay. Then this is something that I call panel plot, which I guess is all known. Is you show the on a given 2D projection. In this is the t-SNE that is coming from the Seurat object. You show the expression of these 5 genes and you can restrict the visualization to any number of clusters or any subs dataset that you wish. And this is another way of right. So here I'm looking at the expression of these genes that are correlated or connected with this pathway that we have identified, and now I'm selecting all the cells that are expressed in a reasonable manner. So I'm removing the high expressed ones because I didn't see really what is going on here, right? The few cells up here, they are disturbing the color coding of the other cells. And so I wasn't really happy with this, and so now I can actually focus on the subset of cells, and it's not that much better, but at least a little bit better. We can now at least see the higher expression or where the individual genes are occurring. And so this is this view is basically shown if you have a factorial on the x-axis and UMI count on the y-axis. And this is, I think, one of the most interesting views for me. It's because you really see the median and the distribution per cluster, and not a distorted, color-coded view. Okay. This is just to show you an example of the heat map and how you can manipulate this. Once you have a trajectory, you can then order the cells by trajectory and look at the expression of individual genes along this trajectory. Visualize the clusters, so you can now see that actually you have I don't know, what is this, cluster 3 and cluster 4 or 5 in the early stages, and cluster 2 in the late stages of this projection, and which correlates obviously with the late stages and of the samples. Right, so I basically we are nearing the end of this talk, and I can show you a little bit afterwards if you are still interested on the live demo, and potentially answer some questions. But what I didn't talk about is, or at least I didn't show you, is the how one saves the RMD files or the images to the RMD files. It's a little bit peculiar because we are saving the function and the raw data, and so this allows you to manipulate the graphs afterwards and optimize them for any publication, but you have them separately. You can bookmark. It's relatively easy to implement new functionality. And since I am more or less the only developer there, I have implemented a lot of tools for debugging, and mostly for because debugging Shiny applications is a little bit challenging at times, and so I'm able to switch on and off this debugging mode anytime and so on. But this we can discuss potentially later, and then didn't talk or didn't show the alluvial plot because I don't know, it's not too interesting. And so I am going to focus in the future on, or at least it's planned, to really modularize the visualizations even more. So and allow for our users in the at Pasteur to use these visualizations in their reports, because so one of the objectives now is to give to our, I call them customers, collaborators, whatever you want to call them, the biologist, something not like a written PDF document, but something that they can actually use to analyze and to look and play with the data that they receive from us. Right? So when they come to the CBU techs or to our platform, they can run the experiments. They are trained in this, and then they usually run the experiments themselves. And then sometimes they have problems analyzing the data, and so we take part of this analysis, take care of this analysis part, but then later on they have to really interpret the data themselves, and for this they actually have to play with the data. And so now we wanted to provide these as modules that can be integrated in RMD files. And yeah, so there are already a few publications out there that using this. Please contact me if you have any questions. And this is the thank you slide with the members or the most involved in this project from the CBU techs. I wanted to mention Michel Pusia, with whom I am collaborating mostly in integrating new features. And I started off with using CellView, which was a very rudimentary, shiny application for single-cell data back in the days. And so thank you for staying with us, and I'm either open for question, or I can give you a little bit of show of the live version, as you wish.
Thank you, Bernd. Do we have questions? I have one question. It's kind of general. How do you work with your user base to make sure that the type of language that you use, like factors, things that are probably not completely common for biologists, how do you make sure that they understand what they are doing?
So now everybody who is entering Pasteur has to, or the new ones, right? They have to go through some training, which includes actually an R course. And to explain a factor, what a factor is not that difficult in the end. And the language, it's for SCHNAPPs particularly. The training is on an individual basis, on a project basis, where I give a 1 or 2 hour introduction into how to use SCHNAPPs with their own data. And then it's a back and forth for them to use it. Yeah.
Okay, and a follow-up question. Are you planning to open this to general users beyond Pasteur?
Yes, it is on GitHub and it's available. So it is open. We are in the process, and we have started already to use something like a light version, which doesn't have the functionality of reanalyzing the data. It's mostly focused on the visualization part. And for the presentation of our data, when we when somebody does a publication with us using our facility, then we encourage them to use this kind of Schnapp slide that we host on a Pasteur server, and then the world can actually play with the data on this server. So
okay, thanks.
Yeah.
Great. Thanks, Bernd. That was really neat to see the live demo, and the Tempora integration looks great because you've added new features that are not part of the R package, the Tempora R package. So I noticed also that you checked in some GitHub code to parallelize the pathway finding. So did you find that you needed that for your integration, or is that just something that you wanted to do?
So I needed, so I'm working with a single cell experiment object. And there is, I don't know if it's a bug or whatever it is, but there was some issue that I couldn't work with the code as it was. And I had to modify it in the end. And I put an issue in there and I forked it, and so I don't know,
Okay, yeah. So I guess it would be good for us to integrate that and test it and integrate it. So anyway, thank you. So yeah, I was just wondering, did you do it because you needed it for speed or something?
No, I mean, so there's one. This was a major, I don't know, change that I had to do, which is just transforming. I think a data frame into a data table or something similar. And there is another kind of improvement kind of thing where I parallelized the, or I don't know, one of the calculations that for the p-values.
Yeah, that's what I'm talking about, the parallelization part.
Yeah,
why you needed to do that?
Because I am using a very large p-value. Yeah, maybe I can show you now this. So here I'm using a p-value of 0.5. This is, has also historical reasons, because one of the pathway databases were not really up to date at some point, and so whatever. But now I am providing all of these pathways with a p-value of 0.5 and smaller in this table here. And which allows us to look for a specific pathway that is not significant, but that we can still look at this, right? I mean, here I can, I don't know, I can take the highest
and
path
search them, yeah. Okay.
And then display this one as well.
Okay, great. Okay, that makes sense. Okay, great. And then do you have? It would be great for us to link the Tempora website to your website, but I guess it would need to be linked to something Tempora related on your website. Do you have a place where you've documented the system and have screenshots, etc.?
Yeah, so on the GitHub
that we could link to?
Yeah, there's a GitHub page with the documentation, but this does not include the Tempora part. I wanted to keep.
Maybe when you release it. Yeah.
I wanted to keep
What's your release plan, I guess? Okay, go ahead.
These contributions in a separate directory. So to keep really the main kind of part from the additional things. And yeah, but we can discuss this.
Okay.
Just let me know what you need, and then I'll figure a way out to do this.
User interface features that other people might like. So we could, if they're coming to Tempora, can you hear me? Because my internet connection is unstable.
There was a hiccup, but now I can hear you.
Yeah, okay. Okay, so yeah, if they come to Tempora, it would be nice for us to tell them about Schnaps and those features that are available, and the easiest way for us to do that is just links to your website to a place that, hold on one second, sorry. So yeah, just a link to a website, and so whenever you're ready, then you can do that.
Yes. I am planning, well, I don't know, I had issues with Docker, and now I am trying out the VirtualBox. And so this allows me to really optimize everything for one platform. And the Docker issue was related to, I don't know, my CPUs are too old for certain compilers. And it crashes with some of the cytometry software that I'm using. Anyway, so now I'm with VirtualBox, and there you just have to download the 6 or 8 gigabytes, and then you're ready to go. Okay,
great. Okay, thanks. Yeah, really cool. Thank you.
Well, thank you.
Also, I don't know what happened, but some people were texting Javier that they couldn't get into the Zoom or they're having technical issues. Hopefully.
Yeah, I don't know what happened today. I saw some of them. I let them come in, but the system was still saying they are joining. I don't know if there have been some
Yeah, I think there were. We probably should have recorded the session or something because usually we have more people, and I think today there's some kind of Zoom issue that some people couldn't join.
Oh, okay.
Technically, some technical reason.
You didn't record this?
No.
Okay.
No, but anyway, we'll send a message out about Schnapps and tell people. Now that we know about it, we'll be talking to people about it. So anyway, it was good.
Yeah, I made this a private recording, which I potentially, with your agreement, put on the internet at some point. Then.
Sure,
That's okay with me.
Okay. So yeah, I don't know. I can distribute the link first, and then I have to do some editing, I guess, as well.
Okay, I think that would be good because yeah, a couple of people I noticed. Some people in my lab couldn't join for technical reasons, so they don't. Yeah. Okay.
Great.
Okay, thank you, Bernd. And thank you, those who joined today.
Well, thank you for having me. And good luck with the pandemic and everything, and Tempora and everything else. Huh?
Yeah, no, it's super great that you've integrated these, and I'm very impressed with the features and like the usable, use cases that you're supporting. But it would be good for us to know eventually, how your collaborator who has the time series data, how they find it. We've been exploring Tempora a little bit more recently with different diverse data sets, and it is. We're still working well, but there's some additional things that we discovered that if you're not using it for a developmental biology data set, if you're using it for another data set, that's time series, but sometimes cells get connected where they're probably not supposed to be connected, and we just have to do some kind of manual curation of the results. But then once we did that, it was providing useful information.
Okay, I was thinking also about the pathways themselves and the individual clusters. So for me, using all of the pathways might not be optimal in the end because some of them are really insignificant. And then I didn't really quite understand completely why all of the clusters have to be included. And I think if you have one cell type that is developing over time, or I don't know, it really depends on the biological question, I guess, but then you are interested in this one cell type and don't care about the other cell types that are in your data set as well, right? So I don't know. There's certain things that I am still not kind of too comfortable with.
Okay, great. Yeah, it'd be good to get feedback in the future once you learn how it's applying to different data sets.
Yeah. Cool. All right. If there are no other questions.
Okay. See you, everyone. Thank you.
Okay, thank you. Thanks, Bart.
Thanks a lot.
Good evening.
Okay. Bye-bye. Okie dokie, duck, OBS.